Yeah, thank you very much, Jürgen, Arnold, and Sergio for the invitation. It's really a pleasure to be at this wonderful site, and I think this is a really great workshop, and for sure a great experience also for you. So yeah, I will tell you about reconstructing human neurogenesis using single-cell transcriptomics. And generally, my lab has an interest in human development. And of course, it's very fascinating how during the course of human development from a single cell, this whole complex organism forms that is composed of many different cell types. And of course, here you just see a tiny selection of these cell types. And yeah, I mean, as you all know, underlying this is the presence of stem and progenitor cells that are that can self-renew and at some point make a decision to differentiate into a particular differentiated cell type. And we are very interested in what genes are kind of driving these lineage decision-making processes, and more broadly, how do different lineages within a complex organ orchestrate their differentiation processes. And actually, is this a pointer? Yes, good. It's a very exciting time, as you saw this whole week, and you probably knew already. It's an exciting time to study human development because we can now model it in vitro. So we cannot just generate distinct cell type through differentiation of iPS cells in vitro, but we can also use the self-organizing capacity of these stem cells to generate three-dimensional tissues, human tissues in vitro. Of course, you have heard a lot about organoids, and cerebral organoids are one of the systems we are using, grown after the Lancaster protocol. And one particular method we are very excited about, as Jürgen mentioned already, is single-cell transcriptomics. And this is a very powerful method because it can very unbiasedly dissect the heterogeneity of these complex tissues. And so we think that, and of course others think that single-cell genomics is, and in particular single-cell transcriptomics, is highly powerful in kind of looking at the cell composition of primary tissues as well as of these in vitro tissues, and by then doing comparisons, we can really assess how well we are able to model human development and also human mature differentiated cell types in vitro. And I will tell you, because originally I was supposed to also give some introductory lecture on single-cell transcriptomics, I will tell you a little bit about that. I know you have heard already a lot about single-cell data. People presented t-SNE plots and these kind of things, but I try to still go a step back and tell you a little bit about, you know, how to do these experiments and how can one visualize them. And please interrupt me at any time, because it's good if you then have this as a background to follow also the other slides. So generally, what is the general workflow of a single-cell experiment? We start with a complex tissue. Of course, it can also be cells in 2D culture, but in this case, a complex tissue. And then we need to dissociate it. We need to obtain single cells. And so we disaggregate the complex tissue with an enzymatic cocktail. This depends on the tissue you look at. For brain, papain is, for example, a very powerful enzyme. But yeah, this can also be collagenases or elastase or trypsin. There are different options. And then you generate a single-cell suspension that in the beginning is still quite crude and dirty. And you want to get rid of any aggregates or doublets or triplets because they will, and also cell debris because that will disturb your single cell measurements. So oftentimes you do filtering to remove multiplets, and you can also do gradient centrifugation to remove debris, for example, cell debris. And then, or you can use FACS to isolate single cells. And then you have a single cell. In this case, this was a neuronal tissue. You can still see the cell still has some processes. And then there's the option maybe to image a cell or not. This depends on the method that you use. And then we lyse the cell. And so now you have the cellular DNA, RNA in the solution. And here for single-cell transcriptomics, you have, in particular, messenger RNA. Yes?
One question. If you want to do it in organoids, do you need to use a special enzyme to deprogram matrigel before? Or
so organoid tissue is a bit more, it's definitely softer than an adult brain tissue, but also the fetal human brain is relatively soft. So sometimes the dissociation even works just with acutase. Or, I mean, actually, we still use papain, a papain-based protocol, which in our hands worked the best. But yeah, you always need to, from the tissue that you have, do several tests. What is the best dissociation method? What enzyme works the best? You want to have high survival and the least number of multiplets as possible. So it's a balance. Yeah.
Do you embed your organoids in matrigel or geotracks or whatever? So like in the protocol in general. So,
yes.
So do you then make them loose from the extracellular matrix before you dissociate them, or?
Yeah, that's a good question. Actually, most experiments that we did were at a stage where the organoid had grown out of the matrigel. We recently did also very early experiments, and there, one can cut the matrigel kind of away. But yeah, I mean, actually everything I will show you is on organoids that have grown out of the matrigel. Yeah.
This process of disaggregation of the cells, do you use the RNA that in the neurites or in the proteins?
Yes, that's another good question. And for sure, is for brain tissue in particular, especially the most mature brain tissue, you have quite some debris which stems also partially from processes that just get, cut off and then the membrane still closes afterwards. And you then lose any messenger RNA that was in these processes. I talked to a person in Berlin that really does, she separates processes from soma and does RNA-seq on both. And you definitely have specific messenger RNAs enriched in the processes, but whenever you have a messenger RNA there, you also have it in the soma, because that's where the messenger RNA is made. I was curious whether there are any messenger RNAs she finds exclusively in the processes, and she says no. It's always, there's way more in the soma, so we are not so worried about it. It might be there are slight differences in abundance that we don't pick up, but anyway, these measurements are, I will say a bit, there's a lot of noise in these measurements because there's very low material to start with. So anyway, you don't want to overinterpret your data and interpret something into a 1.2-fold difference or something like that. Yeah. Yes?
Do you know if free-floating RNA can also get captured and retranscribed? So,
yes.
For instance, cells undergoing cell death, or
yes. So definitely, you do you capture individual cells. If there's now still a lot of RNA floating around, then it depends very much on the method whether you still have that in your experiment and will generate cDNA from it, or whether you can wash it away. And I can show you, because if you capture cells and wash the cells first, you can of course try to get rid of it, unless it's attached to the cell somehow. But if you do the newest droplet-based experiments, then of course you have it in the droplet, and there you want to make sure to have as little as possible free-floating messenger RNA, which you can get by washing the cells more often. And you can definitely, initially we did experiments taking supernatant and checking for free-floating mRNA. Yeah.
Does this dissociation process affect expression?
Yes, another very good question, and everyone that does these types of experiments should be aware that there are definitely changes you introduce through the dissociation. And one good thing is to always, once you find some interesting gene that you want to study in a particular population, to go back and do actually stainings, either for the messenger RNA with fish or for the protein, to see whether actually also the protein is there. Yeah, especially immediate early genes. If you see an enrichment for immediate early genes, you should be careful. This can be something meaningful, but it can really be also a response of the cells. CFOS and these kind of things, which in neurons are of course also interesting, but it could be an artifact. And there are some genes that we see maybe across different tissues, because we don't just look at neuronal tissues, and that we see also in other tissues. And then even more we are careful that that could really be just something due to the dissociation. I think at the end it's always good to do a hybrid between, you know, these dissociation-based methods and also do something in situ. Yeah. Okay, let me move on. I might get out of time at the end. So then you have this initial messenger RNA, and you always need to amplify your material. It's way too little to detect. So you can amplify the cellular RNA, and there are two different ways, or broadly two different ways that are currently used. Either you do reverse transcription and PCR, or you do reverse transcription introducing a T7 promoter and then doing in vitro transcription, and then generating from this amplified messenger RNA the cDNA. So it's the in vitro transcription way is a linear, a quasi-linear way. The PCR method is an exponential way, which introduces a lot of bias. But one can circumvent this bias by introducing in the very beginning unique molecular identifiers, so-called UMIs, which if in the very first step of reverse transcription you introduce such a barcode to every unique transcript, a unique barcode for every transcript, then you can amplify as much as you want. At the end, you will be able to track all the amplified molecules back to this one initial transcript. So this is a way that most protocols nowadays use. And then you prepare sequencing libraries. Also at that step, you add barcodes. So these barcoding things are very important, because then you can multiplex many libraries from many cells, and you don't have to sequence one library per Illumina lane, which would be extremely expensive, but you can multiplex hundreds and thousands of cells per lane. And then you do computational analysis. And so generally, so you get sequences from the sequencer, you align the reads to a genome or a transcriptome, then you take an existing annotation to say what genes are actually expressed. And what you end up with is a matrix where you have genes, for example, in rows, and cells, for example, in columns. And depending on how many cells you have, well, genes in the human genome, maybe 21,000 genes, even more if you have isoforms. And then you have maybe a couple thousand cells. So you have really a matrix of a couple thousand columns times 21,000 rows, which is a really big matrix. And to make sense of that and to really identify particular genes or cell clusters, you have to use statistical methods. You cannot look at this in an Excel, cannot open the whole thing. So there are always, before I go into that, I mentioned already, but challenges of these experiments are that you have to isolate a single cell in the beginning. It's very low input, so let's say 10 to 20 picograms, but this can be even less, or it can be also more, it depends on the cell type. For messenger RNA, actually DNA is even less, you have just 2 copies, right? Unless you go for a dividing cell and you have maybe 4 copies. And then you have biases that you introduce during amplification. I mentioned you can use UMIs to try to get around that. If you bring in contamination from the free-floating messenger RNA or just from the experimenter or from some previous experiment, you have a problem. You won't be able to distinguish them. And you have a lot of noise that is technical due to the low abundance of these messenger RNA, but also biological noise, which is meaningful, and it's hard sometimes to distinguish one from the other. And so now I mentioned one challenge is isolating cells, and there are different ways to isolate them, and there are more ways than just these 3 ways, but these are the ones we are using, and I would say these are the ones that are most commonly used. So this is a method, this fluid MC1 system, probably everyone knows, but this is also how I started to work with single-cell transcriptomes, because this is a method that was really pioneered in the Quake lab where I did my postdoc. It's valve microfluidics based, so you have this chip in the middle, and there are these chambers that can be isolated and where flow can be pushed through, liquid flow and flow of liquid with cells, and you are able to capture individual cells. Here is one cell in these little nests in these chambers. And here you can then wash over the cell, so you can get rid of free-floating messenger RNA. this is one way. It's a little bit, it was high throughput maybe 4 or 5 years ago, now it's low throughput. Then in the middle is a quite versatile method where you just fact sort individual cells into individual wells of a multi-well plate, 384, 96, whatever. You can even use robots to then process and generate the cDNA from the cells. And of course here you can have transgenic labels or antibody labeling and then sort specific cells. So it's very versatile. It's a little bit more in the middle throughput. Of course, if you use robots, you can get thousands of cells, but it's a bit more tedious than these droplet microfluidic-based method, where here you don't isolate cells into really a compartment that is static, like a well or one of these chambers, but you isolate cells in aqueous droplets, which are, again, it's a compartmentalization of the individual cell. And the trick of these methods, which is drop-seq, in-drop-seq, developed by InDrop by Alon Klein and Linas Matsoutis, or DropSeq, which was developed by Makosko et al. In the RegF lab. And also more recently, 10x Genomics is a commercial way of doing these kind of experiments. The trick here is that you don't just isolate a single cell in a droplet, but also you add in a bead. And this bead contains the primers for your reverse transcription. And every bead has a different barcode on every of the primers. So at the end, if you have one cell with one bead, this cell will have a unique barcode for all its transcripts. And then you have another cell with another bead. Again, every mRNA of this other cell will have yet a different barcode. And these cellular barcodes really enable very high throughput experiments. And actually, you can think this even further, and there are even newer split pool barcoding approaches where you don't even have to physically isolate cells, but just by combining different barcodes and splitting cells, combining them again, splitting them again, and in every splitting step giving them a barcode, you can also in that way demultiplex your single cells at the end. So this is very exciting, and this is, has been a very fast moving field. And so now, when you have this data, as I said, this big matrix, what can you do with it? How do you make sense of it? So the usual first step is principal component analysis. And here I'm just showing for some data, PC1 versus PC2, so principal component 1 versus 2. And principal component analysis is a linear way of reducing the dimensionality of your data. And in principle, you undergo orthogonal transformations of your data. So you look in the first principal component, what is where is the largest variance in my data set? And the second component is orthogonal to the first, and it gives you the second most abundant variation in the data set. And the way you can think about this is that every principal component will identify genes that are somehow describing variation in your data set. So in this case, you can now see what genes are actually driving this principal component first, which show you that these genes really distinguish these cells from those cells. So in principle, with every PC, you identify really meaningful genes. Principal component 2 identifies another set of genes that actually, in this case, only these cells here on the right, on the left get split up. These cells are independent, they are at 0. But these clusters here that were clustering together, if you just look at PC1, they cluster now separate on the basis of PC2 because of these genes. And in principle, this is now just giving you PC1 and 2. Now you could go further and look at PC3 and PC4 and always ask, what cells get now separated on this plot? So PC1 was these green ones from this dark turquoise and blue. PC2 was blue from the dark turquoise. And now you can move on, and PC3 will probably also distinguish some cells from each other and identify genes that drive that. And so it is a very good way to identify genes that are meaningful for clustering your cells. It is not necessarily a good way if you have many different clusters available to visualize them in 2 dimensions. because here we can only visualize PC1 versus 2. And everything else you would need to visualize yet with another 2D plot. So this is where t-SNE comes in, t-distributed stochastic neighbor embedding, where you can now use the genes that you learned here, maybe even for that are meaningful for the first 10 PCs or first 20 PCs, to then cluster in a non-linear way all the different cells that you have, all the different populations separate from each other. So in this case, you then really take all these gene sets and just ask which cells are similar, which cells are different. And the similar ones will cluster in a group and will cluster away from other cells that are different. So here you cannot say anymore, this group is closer to that one than this group is to that one. This you cannot say. It's really nonlinear. It just means this one is different from all the others. This group is different from all the others. Yeah, this is how to read these t-SNE plots. They are super powerful if you have many different clusters. And then there are other ways to look at your data. You can also do cell-cell correlations. So you look at the transcriptome of one cell and the transcriptome of another cell, or for each cell, the subset of these PC-identified genes. And say, how is one cell similar to the other? You can just correlate the cells. Some cells are highly correlated, and these cells might then be not correlated at all with another group of cells that are then by themselves again correlated. So here you can also see clusters in these correlated squares. Or you can just plot a simple heat map. After identifying all these gene sets, you plot a heat map of all cells and all genes, and then, no, actually, cells and genes this way. And then you see what there are different cell populations. And generally, the way you now say this is a cluster and that is a cluster, that's yet something else. You could do it on the basis of these 2D projections, but better is to do it based on the transcriptome level. And you can do k-nearest neighbor clustering, hierarchical clustering. There are all kinds of different ways to cluster to then really say, actually, within this cloud, there are 3 different clusters. These kind of things. And I would suggest you, if you are interested in these type of analysis, to check out the Seurat package from the Satija lab. This is really a very easy to use tool for single-cell RNA-seq exploration, and it also comes with, you know, exemplary data that you can download and play around with. Yeah?
Yeah, sorry, I have a question about the t-SNE. So if I understood it from my notion, I have also this k-nearest neighbor, or this index is at the end also a bit arbitrary, so you can decide this index.
Yes.
So do you have like, especially from this kind of data, way to decide what is the meaningful k-index to define clusters?
I mean, there are ways to try to figure out how many clusters might I have. But at the end, you have to personally decide which set of parameters was now best, how many clusters do I have. It is nothing deterministic that will be told you fully unbiasedly. So in this case, it's always an iteration, this whole analysis. So you have something, you look at it. What we always do is then we look really, what are the genes that mark these clusters that I now have? And if you have the feeling that this is not really a clear cluster, it's just, I mean, it can be just one gene is different. This might be meaningful or it might not be meaningful. Or if it's a big group of genes that are differentially expressed, then I usually take it as a meaningful cluster. So it's always, yeah, iterations. And finally, how to visualize expression now in the populations. One can take these t-SNE plots and just project the expression of genes on them. This is very straightforward and easy to understand. One can generate violin plots which still show you a distribution of the cells in each cluster. So here are, for example, 3 clusters, and then you show the distribution. So here, down here is 0 expression, and then the expression goes up towards the top. And in principle, it's a histogram that is put on the side and mirrored. So if you take a line through here and turn it 90 degrees to the right, you just have a histogram. And it's just an easier way to visualize, to understand histograms. So for example, this cluster has high expression of this gene, and this cluster 2 has low expression of this gene, G5. Yeah. Then dot plots is another way. In this case, the fraction of the cells expressing a given gene is the size of the dot. So a big size means pretty much all cells express it. Small dot means almost no cell expresses it. And then the level of expression, the mean level of expression, is then the color. So you see here are, for example, 2 groups of clusters. And then you can generate bar plots. You just take the mean of expression in each cluster and some standard deviation, and then you get a plot like that. So then something else that you might see in the data, and this is mostly relevant for all these developing systems, is that you don't necessarily have very clearly separate clusters, but you have some gradient between them. So in this case, you have cells here and genes here, and you can see that there's some overlap of gene expression in this middle here. And this shows you that there's actually a transformation going on. These cells can transform into those cells. And when you have such scenarios, you might want to infer some trajectory of the cells. And if you want to infer that, you can use different tools. For example, monocle. This is monocle 1. There's also monocle 2 available. This is all developed by Cole Trapnell in his lab, where you project the cells similar to PCA in 2 dimensions and then plot a minimal spanning tree to connect the cells in this kind of lineage. There are diffusion maps, which kind of work with random walk analysis of the cells, in this case coming from here and then going in 3 different directions. You can use Spring. It's developed by the Klein lab, Alon Klein's lab. It's a way of visualizing, it's a graph abstraction of k-nearest neighbor, k-nearest neighbor clustering. Then you can do intercellular correlation networks, ask how similar are cells, and if they are similar, you put them next to each other in such a network. And most recently, for example, this is a very clever trick, is RNA velocity, developed in Stan Linneson's and Peter Karchenko's lab, where in this case, this is, for example, a Tiersny plot. You can ask, and this goes into detail of the single-cell transcriptomics, where if a transcript is made in the beginning, it still contains introns, and those introns get spliced out. So based on the availability of introns in a given gene, and then the absence of the introns in the same gene, you know some directionality of the cells. You know which cell came earlier, that one still had the intron, and which cell came later, it lacks the intron. And then you can, I don't know if you see, but you can make little errors on your plot, and you see what is the direction of the transformation you are looking at. So now I'm already at half an hour, but I want to go back to this initial slide and say, okay, I was telling you that we think single-cell transcriptomics is really powerful to compare primary tissues and in vitro systems, and I want to tell you now some work from my lab. The first is focusing on cerebral organoids and analyzing them with single-cell transcriptomics, and the second goes into reprogramming of somatic cells into induced neurons. So in the beginning, when we started using cerebral organoids, and we actually, one of my students learned from a student in Wieland Huttner's lab who had learned from Madeleine in Jürgen's lab, so we learned how to grow them. And we were asking the question, how similar are these organoid cortical regions really to the fetal cortical regions? On the basis of the staining, they look very similar. And I don't have to introduce anymore the architecture, the layered architecture of these regions. So we have progenitors and neurons, progenitors apically, neurons basally. But we were wondering on a genome or transcriptome level, how similar are they? So we performed single-cell RNA sequencing on both the fetal tissue and the organoid tissue. And this is now the data on the fetal developing cortex, where we knew very precisely that we are looking at cortical cells because we microdissected them, or actually Marta Florio in Wieland's lab did that. And we do PCA to identify genes, just like I showed you, and now these are the genes that we identify by PCA, shown in a heat map with cells in rows and genes in columns, and everything hierarchically clustered. And you can see that there are different groups of cells. So warm color is high expression, blue is no expression. And you can see the presence of some genes that are really known genes, so they are marker genes for different populations. And up here we can look what these gene clusters are enriched for, what the genes are associated with doing geo-enrichment analysis. And if we now put all this information together, we can, one additional thing, you see this sidebar here, which are these different colors. So since we had isolated the cells after dissociating the tissue, we didn't know anymore, was the cell actually in the VZ, in the cortical plate, where was the cell? And so what we did here is we calculated the correlation of each of these individual cells with bulk transcriptome data of microdissected regions from the human cortex. So the VZ, the ISVZ, OSVZ, and cortical plate, which is from the Feeds et al. PNAS paper, these bulk transcriptomes. And then you can see that high correlation in red means that these cells very likely derived from the VZ, the ISVZ, OSVZ, and the cortical plate. And we actually have a nice representation of all these different zones. This is, of course, just an approximation, but it gives us some way of trying to label the cells with a region. And then we can go into the details of what cells we are having here. And we have apical progenitors, which actually contain this group of genes, including PAX6. They are highly proliferative, MKI67. And they also have this gene cluster, which contain SOX2 and vimentin. And then there are basal progenitors here, which actually are very specifically marked by EOMES or TBR2, and they lack these genes here that are only found in the apical progenitors. And then we have a big group of neuronal cells that all have these neuron differentiation-related genes, but there are subgroups here which we found very interesting. One subgroup here, they express genes involved in cell migration, which was very cool to see because, of course, once the neurons are born, they have to migrate up to the cortical plate. So we think these are the newborn neurons. They are still migrating. And then here are more mature neurons that establish already in the cortical plate. And then, just like I showed you, we can, because here are gradients, we can infer the lineage trajectory. This was done here using monocle 1, starting here from apical progenitors through the basal progenitors to these early migrating neurons and the later more mature neurons. And this is then, when you now plot a line through here, you can say this is my pseudotime, my differentiation pseudotime, and you can show how the genes are expressed according to this pseudotime. And I must say this is gestational week 12, 13, so this is still very early during cortex development. We don't really see outer radioglia at this stage. Yeah.
Is this a single line, or can you integrate multiple lines in such a plot?
So in this case, this is a single line, but you can have branching off of other lineages. For example, this, what I showed here, this is, for example, also data from my group. It's a completely different system, but here we have progenitors that really generate 3 different lineages. And the same can be true for monocle plots. Actually, I will show some monocle plots also with branching pattern. So, was there another question?
Yeah. I have a short question. For this analysis, did you took the entire organoid and dissociated, or did you?
This is still the fetal cortex. So here we know what we have. I will get to the organoid pretty much now. First, I show you that we can also just see how are transcription factors expressed. Of course, there's an interest in transcription factors and how they might determine the fate of cells, and we can generate these networks for them as well and say dots are individual TFs, and you know the transcription factors that are nearby and highly connected are very similar in the expression profile. So we see here a progenitor subnetwork. Then we have factors that are very important for the transition for these apical radioglia to basal progenitors. Here's TBR2, insulinoma 1, and then we have the onset of this neuronal program and then a neuronal subnetwork up here. And so now we move to the organoid. And so here we did experiment on whole organoids, but also on microdissected regions. So we slice organoids live with a vibrotome, we identify cortical, like ventricles, and then cortical areas surrounding them, and then we microdissect them, and then we dissociate these. And I must say, this is still fluid IMC1 data. So this is, we published this 2015, so it's a few years ago. And when we did now PCA to identify genes, and then t-SNE to visualize them in 2D, the cells, we see that there are different clusters. And we can now look, as I showed you, we can project the expression of genes on these clusters to identify what they are. So we have actually neuronal cells in these two clusters. They express all MID1-like, for example, as one neuronal marker. And then we have proliferative progenitors here and there. Now still the question is, what kind of maybe regional identities do we have in these organoids? And here, FOXG1 is really a marker for forebrain, but we want to distinguish dorsal from ventral. And we know that OTX2 is expressed in the ventral and medial cortex, but not in the dorsal, not cortex, forebrain, but not in the dorsal forebrain. And so we really look for these FOXG1 positive, OTX2 negative cells, which we find in these two clusters. So we have cortical progenitors and neurons. And here we have ventral cells, as well as really earlier cells that came from 30-day organoids, for example. And interestingly, since we had these microdissected regions, we were wondering, where are these regions found in this plot? And you can see that 3 regions, this one, 1, 3, and 4, are all found in the cortical clusters across progenitors and neurons. And then region 2 was actually a ventral forebrain region. It is in the other cluster. And we can really see how nicely this is segregated. So if you have such a nice region, it's very likely just one regional identity. Yeah, this shows just distinction of NeuroD6 versus GAT1 in these different regions we dissected. And now we want to compare these cortical cells that we identified with our primary cortical cells from the fetal cortex. And we do this by putting all the cells together and doing this intercellular correlation network analysis. And now you see here color-coded by cell state or cell type that we identified from the apical radioglia through basal progenitors or intermediate progenitors to neurons. Also here color-coded by these marker genes. And now we ask which cell is a fetal cell and which cell is an organoid cell. And you can see that there's nice mixing of these cells within the network across both the progenitors subnetwork and the neuron subnetwork. And this shows us that these cells, once we identified which ones are the cortical cells, are actually highly similar. We can also do a comparison on the gene level, so for example here ASBM, and how it behaves over pseudotime in the organoid and primary tissue, and this is very highly correlated. And so this made us actually very confident about these organoid systems, but it also makes us aware that there is more in the organoids than cortical cells. So just doing bulk transcriptomics on an organoid is really not a good way, because you might have different proportions of regions. And this we now know even more since we have moved on to do high throughput experiments on these organoids. And here, if we, for example, look at a single organoid, we can at once see all kinds of different regions that are present in the organoid. So here, this is 4,900 cells from a single organoid. And you see how we have progenitor cells, and we have the dorsal, the cortical lineage emerging, the excitatory neurons here labeled, for example, by NeuroD6. But we also have a ventral lineage marked by the DLX genes and GAT1. We have proliferative dividing progenitors. We have mesenchymal cells, which we actually had before as well. I didn't mention it. They are in the minority. And we have here also some cells that have a hindbrain phenotype. And actually, we have done these high throughput experiments now for many organoids, actually 23 organoids from 7 different human pluripotent stem cell lines, 1 ES line and 6 IPS lines. And so what we do here is we identify what are the progenitors, we identify what are the neurons, this is a t-SNE projection, and we can now based on similarity to the different brain regions. And here we take as a reference the Allen Brain Atlas transcriptome data from many different regions and different developmental stages. We can say what we have here. And actually here on the left, you have really all the cortical cells. And then on the right, you have all the subcortical cells, ventral forebrain regions, as well as midbrain and hindbrain cells. So cells that really strongly similar to cerebellum, for example, or to the midbrain. But if we now look at this projection for different lines and different organoids, so here are now 2 organoids from 4 different lines, a subset of our data, and you can see how some organoids make really a good mix of cortical and subcortical regions. Other organoids make really pretty much only cortex. I mean, there are a few other cells, and other cell lines make, or not even cell lines, other organoids, for example, this here was really no cortical region found, whereas another organoid from the same line made also cortical cells. And one can summarize this and just ask, what is the capacity of each organoid to make these different regions? Then you can see that there's high variation, and this is very consistent with what Jürgen's lab also published, the Renner et al. EMBO journal paper, that there are differences. Actually, these, for example, are 2 different batches, where in one batch you have much more abundance in midbrain, hindbrain regions, whereas the other batch had pretty much just dorsal and ventral forebrain. We can now also use this data from all these lines and all these organoids to reconstruct differentiation pathways. And here we use Spring, which is really powerful since we now have 3 different lineages present. Or actually, it should be 4, but midbrain, hindbrain, so far we only see kind of as 1. It doesn't fully segregate out. But we can now here, for example, show this Spring plot with color-coded based on how similar they are to our reference, the Allen Brain Atlas data. So here we have the cortex, ventral forebrain, midbrain, hindbrain lineage, and we can show also expression of genes. NeuroD6, for example, very strongly in these cortical neurons, DLX5 in the ventral lineage, and TFAP2B, which is a cerebellar marker. And finally, also here we can say, okay, we have the dorsal forebrain lineage. Now, how do certain genes, are certain genes expressed based on pseudotime? Yeah, and so taking all this together, we were very confident about these organoid systems in that they recapitulate gene expression programs of early fetal neocortex development. And now we can use them to ask, for example, evolutionary questions, and you saw some of that already by Wieland. And also we can use it to model disease. And I will show you a little bit on both these areas. So Wieland already introduced this, but we grew organoids not just from humans, but also from chimpanzee. On the level of these stainings, these look highly similar. Also, if we now do single-cell transcriptomics on the chimpanzee organoids, here's again C1 data, fluidigm C1 data. We can see that also in these chimpanzees, we have VZ correlated apical progenitor or ventricular progenitors. We have intermediate progenitors and neuronal cells. And of course, one thing we were curious about is how, if we now put these cells together, just like we did for fetal and organoid cells, what if we put chimpanzee and human cells together in a network, in such an intercellular correlation network? And here we see something a little different. So now the 2 shades of blue are the human, fetal, and organoid cells. They are still very intermixed. The chimpanzee cells segregate out. They are still spread across the progenitor and the neuron subnetwork, but they are segregated out. So there are definitely more differences between the human and chimpanzee cells than between human, fetal, and organoid cells. And of course now there's the question, what is different? And yeah?
I was wondering, so you showed us how to validate that your human lines recapitulate the development in the organoid, and I was wondering, can you say anything about how to do that in a chimp?
Yeah, yeah, that's a good question, and I don't think we can do that for the chimp. I mean, you would need to be extremely lucky to get to some fetal chimpanzee tissue, but we are not even trying to do that. I think we take the similarity between human fetal and human organoid tissue as an indication that chimpanzee cerebral organoids should model chimpanzee brain development. Of course, it could be that how we, the culture conditions we give to the cells are maybe would need to be adjusted a bit for chimpanzee. And we definitely see that the chimpanzee organoids, it's a bit harder to grow them. There's a bit more batches that fail, also even on the IPS level. And this is the same, for example, for Rusty Gage's group. We talked a lot about that. Also, they have problems with the chimpanzee IPS cells. They don't grow as nice as the human lines. But you can still, you need to do a lot of picking, clean up your culture, and then it still works very well. Yeah.
Yes, sir, just quickly. In this case, it's also micro-dissected cortices from each
yes.
organism.
In this case, and I don't show you here the analysis before to identify the cortical cells. So actually also here we have other cells than cortical cells. We have mainly dorsal and ventral forebrain. This is just the dorsal forebrain cells. Exactly. And so I actually won't go much into the differential expression analysis. So we had an initial analysis here. There are some genes, for example, SOX2, NeuroD6, they are highly similar between human and chimpanzee cells, but there are also some genes, integrin beta 8 and INSR, that show differences. For example, in chimpanzee, the level on the progenitor, in the progenitors is much lower than in the humans. Here it's the opposite way. It's chimpanzee has much less or pretty much no expression in the neurons, whereas the human cells have it. These are now just really two examples, and we are still working on trying to fully get a good list of genes and that are differentially expressed. And here, of course, we now move to doing high throughput experiments where we can really learn more about a single organoid because we have thousands of cells from a single organoid. Also, we have now more chimpanzee lines. Originally, it was just one line or two lines. And we have an outgroup, which is macaque, also orangutan. And so this is just really a teaser. Yeah, we have now data from these species, and we are trying to make sense of it and come up with a list of differentially expressed genes that we are highly confident about. There are many problems with these type of analysis because the genome quality is not the same for these different species, the annotation is not the same, and you can work with consensus genomes, but then you really lose information about human-specific or chimp-specific genes. So there are also here you need to do many iterations, try different alignment strategies, try different annotations, and these kind of things. And at the end, you need to look through gene by gene, what you identify as differentially expressed, to really the raw data, look at the coverage and make sure that it's no artifact of the genome quality or something. Okay, so actually, here it shows 14 minutes, but I'm way beyond 14 minutes. How much time do I have left?
35.
Okay, thank you. So now a little bit on this disease aspect. So here we started by teaming up with Silvia Capello's lab at the Max Planck Institute for Psychiatry in Munich, and we are looking at malformations of cortex development, in particular periventricular nodular heterotopia, which is a malformation that Silvia had been interested in already during her postdoc with Magdalena Götz, and they had collaborated with Stephen Robertson in New Zealand to identify two genes, FAT4 and DAXes, which are actually, interestingly, receptor ligand pairs, protocaterins, that when mutated so either one of these can be mutated, and it leads to periventricular nodular heterotopia. And the malformation that that you can find here is that most neurons migrate in the right way, in this case to the cortex, but there are some neuronal cells that fail to migrate in the right way, and they are in these heterotopic loci stuck at the ventricle. And so what is an obvious thing to do here is to get fibroblasts from such a patient, FAT4 or DAXIS patient, and generate iPS cells and then grow organoids from them, as well as for control patients. And we were very excited when we saw how these organoids develop, because whereas here in a control organoid in the MAP2 staining, you see that the neurons are all in the periphery, located basally. In these mutant organoids, not in all ventricular regions or all cortical regions, but in many of them, you find that neurons are clustering at the ventricle and fail to migrate. This is actually a DAXIS mutant. This is a FAT4 mutant, where even more you have a lot of disorganization, and here are two of these foci with the neurons that fail to migrate. And here's a quantification. So generally, we see quite a lot of disorganization and a lot of this heterotopia phenotype. Now, of course, the question is how are these cells defined on a molecular level? What do we find if we perform single-cell transcriptomics on micro-dissected cortical regions from these control versus mutant organoids? And here we used fax-based single-cell transcriptomics. So we sorted cells in 96 well plates, and then we generated transcriptomes. And generally, if we just look at a set of marker genes for progenitors and neurons, we can see overall we have progenitors and neurons in both cases, control and mutant. It becomes obvious that there are more neuronal cells in these mutant organoids, and this is across two batches and across different organoids. We have consistently a larger number of neurons than progenitors at exactly the same time point, which is 60 days. So it seems like the neurogenesis proceeds faster in these mutant organoids. But now we were curious how, if we put mutant and control cells together and do such a lineage reconstruction. And what we find is that here's now a monocle 2 plot trying to infer the lineage. And you can see, again, here is also a branching or maybe even two branchings occurring. And if we now color code this based on the expression of some genes, PAX6, for example, and STATMIN2 for progenitors and neurons, we see that there is this path from progenitors to neurons that are found in control and patient-derived organoids. But then there's the side branch that is only found in the mutant organoids. And yeah, so you can here see, again, the separation based on whether it derives from a control or a mutant organoid. And it's very striking that this side branch only exists in the mutant organoids. And this is 3 to 4 organoids per, so 4 organoids for both these and 3 organoids for the control, actually. And we see it in every organoid. So now we can segregate this, this inferred lineage into different segments, and we can say we have progenitors, we have this normal neuronal state, and we have this altered neuronal state, and ask what genes are differentially expressed. And very interestingly, genes that are upregulated in the altered state versus the normal neuronal state, or that are downregulated, they really have, are associated with axon guidance and neuronal migration. Also a lot with kind of synaptic, some of them are synaptic genes that is found in the normal state but not the altered state. And interestingly, some of them are associated with epilepsy, which is one of the phenotypes of the patients. I don't know what it means that one of these genes is actually upregulated and the other two are downregulated in that step, in that state here. But still, it's very interesting. The genes that come up are somehow making sense, but we are still trying to fully understand this whole phenotype. And we also did migration assays in 2D culture, where we can just follow, if we generate cortical neurons in 2D culture, we can follow neurons over time and see how they migrate. And we can quantify velocity, resting times, and tortuosity of these migrations. And we can see overall that these cells, the mutant cells, overall on average, they migrate less fast, they rest more often and longer, and they migrate less directed. And one can take these measurements together and cluster the cells. And then, just like one can do for single-cell transcriptomes, you can see that there are two populations that are found in all conditions, but then there's this one population that is found in the mutant organoids and not organoids, in the mutant neurons, but not in the control neurons. And so, of course, we haven't yet fully brought this together, but we identified this molecular signature, this altered neuronal state, of this neuronal state that seems to have a defective navigation system. And it's, of course, one would want to say that these cells are actually these that have this altered state. And we are still, we actually recently performed single-cell transcriptomics on the 2D cells to try to see whether fully cell autonomously, because here they are not embedded in a tissue, not in some microenvironment like in the organoid, whether even there they acquire this altered state. And so with this, a summary on this organoid part, I showed you that cerebral organoids recapitulate gene expression programs of human fetal neocortex development. We can use single-cell RNA-seq on cerebral organoids derived from patients with heterotopia, and this provides some insight into the disease-associated neuronal state in these organoids. And finally, we can compare human and great ape and other great ape organoids and try to identify human-specific features of corticogenesis. And now, in my last minutes, I will tell you a little bit about how we use single-cell transcriptomics on two-dimensional cultures where we try to reprogram a somatic cell type into a neuronal cell type. Yeah?
Pushing to the last part, out of curiosity, I mean, I understand the power in this comparative analysis and this data is incredible. What's the gap? Is there a number on the gap between transcriptomics and proteomics? Because not every mRNA that you find there actually becomes a protein.
Yes.
And even if so, not necessarily at the time point when you are looking, and I don't know.
Yeah. So actually, this is a good question. It would be, of course, great to know the proteomes of all the cells as well, because they are, at the end, the meaningful molecules in the cell. And what one should see here, that the single-cell transcriptomics is really a way of phenotyping cells. And even if an RNA would not give rise to the protein, it would contribute to the clustering of these cells, and it would still be a meaningful measurement. I just cannot make the statement that any RNA we detect will be a functional protein at the end.
So are there attempts to quantify this, just to get an idea?
Yeah, I mean, there are, for example, G and T-seq. There are different ways of measuring proteins and RNA from the same cell. There is also starting to be, ways to do proteomics on single cells. Some of the genes correlate perfectly, and others don't correlate at all. I would say the majority of, more than half of the genes, or of the proteins, will correlate with the RNA, but there are definitely cases where the RNA doesn't correlate with the protein abundance. And of course, even more, we don't know at all about post-translational modifications. So I would always be careful if I detect a gene that I'm interested in. One should do stainings to make sure the protein is there, and then of course look more into the protein biochemistry, the protein behavior in the cell, if one really wants to tell a story about the protein at the end. In our case, we use the transcriptomes more as a way to cluster cells. And of course, if we now see something differentially expressed, it is meaningful for us. It means this messenger RNA is here and not there. Whether it then means the protein is here and not there, we don't know. Yeah, but it's a good point.
So So the separate branch that you find in the disease organoids, can you extrapolate that to the possibility of a complete separate lineage of differentiation?
Yeah, I mean, at this point, this is just an inference. So we of course don't know. It could always be the cells go into that state, they come out of it and go on. We don't have directionality. We don't know about reversibility of these states. And we are still trying to use genes that we identified in that altered state to also stain the tissue to see whether it is in these spots, in these foci of neurons that fail to migrate, for example. That's, of course, a very interesting point, a very obvious thing we have to show. Yeah, I mean, there's a whole set of novel single-cell genomic tools that I would have touched on in the single-cell transcriptomic methods talk, which combines measuring lineages directly through DNA barcodes and transcriptome measurements. And you are maybe aware of these papers that just recently, some papers came out on zebrafish, really beautiful work from Alex Shear's lab and Alon Klein's lab. And yeah, so one would need to do that in order to really know the lineage. Okay, so a little bit on that. I have maybe 15 minutes or?
Probably. No,
10?
23 minutes.
Perfect, good. Yeah, so this is an interest I started during my postdoc when I actually collaborated with Marius Wernick to analyze fibroblasts to induce neuronal reprogramming. And generally, here I show you this network that I showed you earlier, which shows the transcription factors that are being expressed differentially during cortex development in the human fetus. And this is generally the transition from these neuroprogenitor cells to neurons. But it is interesting to me to ask how transcription factors induce generally neuronal identity. And this can start even from a fully independent, different somatic cell type. And it has been showed, especially Marius Wernick has been pioneering this, that you can express certain neuronal transcription factors in fibroblasts and make neuronal cells. And so this is, of course, very interesting, very remarkable transformation of cellular identities. And it tells you also a bit about plasticity of cells. So these cells are actually not stuck in their identity, but you can still modify their identity. And so there are two systems. One is this, what I mentioned, the system that Marius Wernick developed and where I had collaborated with him. And I'm not talking about this. This was published in 2016. You can read more about it in this paper. And more recently, so this was mouse fibroblasts to neurons. More recently, I have been collaborating with Marisa Karov and Benedikt Berninger to analyze the reprogramming of human parasites to induce neuronal cells. And these parasites are primary adult brain derived. So this is a very interesting system because this is a mature human brain derived parasite that then can give rise to an induced neuronal cell. And so this system was introduced by Marisa Karov in 2012, and we actually now, this paper is in press right now. This will soon come out in Nature Neuroscience. Interestingly, whereas here for the fibroblast to neuron conversion, you use ASCL1 or MESH1, which is really a pioneer factor, together with two neuronal factors to make very efficiently induced neuronal cells. You can also use ASCL1 alone, and it still makes neuronal cells. Less efficiently, but it still works. What you need in the parasites, you cannot use these, you cannot use ASCL1, so you cannot use this combination. It won't give you neurons, but you can also not use ASCL1 alone, but you have to use ASCL1 in combination with SOX2, which is very interesting. So one factor is in common, but it needs other cofactors. And this already tells you that what you learn from the reprogramming of MAFs doesn't necessarily tell you that this will apply also to other somatic cells, especially mature cells such as these adult brain-derived cells. And so what we did is we analyzed different time points along this reprogramming path from day 0, which are the human parasites, to day 22, which are these human-induced neuronal cells. And interestingly, these cells are not excitatory, but inhibitory cells. They are GABAergic interneurons, so IGN cells, induced GABAergic neurons. And as I mentioned already, ASCL1 alone does not manage to reprogram the cells, SOX2 alone also not, and it's the synergism of ASCL1 and SOX2 that is required to make these induced GABAergic neurons. So when we did single-cell transcriptomics, we do PCA and then t-SNE visualization of the data. This is what we find. We actually find some clusters that are kind of alternative fates. They are not really wanted. They are not neuronal cells. There's something else emerging, and we had learned that already in the analysis of fibroblast 2 neurons, that alternative fates can emerge during these reprogramming processes. And then here we see a marker for parasites, PTGF receptor beta. It is found in these 2 populations at day 0. And we have neuronal cells here at the tip. And this already shows you that actually there is heterogeneity in the initial population of parasites. We have 2 separate parasite clusters, and only one of these clusters can give rise to neuronal cells. So here we have a productive reprogramming, but then here we have cells that fail to reprogram. And this is a very interesting scenario because it tells you that there are, even in this group of parasites, they all express PTGF receptor beta. There can be a state that is facilitating reprogramming, whereas the other state doesn't. So here's this group 1 that cannot be reprogrammed, and the group 2 that can be. We still don't know whether these groups can interconvert, but that's another interesting question. But we can ask what are the barriers in this group, or what facilitates in that group to allow for the reprogramming. And one interesting thing is that SOX11 is expressed in these cells and not in these. And it could well be that SOX11 as a pre-existing factor is really important to allow these cells to go into this neuronal state. And we can now use only this productive reprogrammings and only these cells that are part of that cluster to infer the lineage, just like I showed you before here using monocle 2. And we have now here parasites at day 0 and how they transition to this induced GABAergic neuronal state. We also can look how different genes are expressed along the pseudotime of this reprogramming process. And you can see how these parasite markers, PTGFR beta, CAF1, CFH, decrease during the process, and neuronal genes, interneuron markers, CHD7, DLX5, and SNAP25 as a general neuronal marker increase. So you go from a parasite state, you lose that state, and induce a neuronal state. Yeah?
Have you tried to add SOX11 in your reprogramming cocktail?
We haven't yet done that, actually. Yeah, we are trying to also knock out SOX11 to see whether then we inhibit the reprogramming in general. It's very hard to mess around with these primary parasites. You cannot really do anything they are hard to modify. But of course, we could yet add a third factor to be co-overexpressed with the others, and then try to see whether then all cells can give rise to the neuronal cells. What we did do is in both groups, in both these parasite groups, we find cell surface markers, and we can separate them via FEX. And then we can really show the one group separated by FEX doesn't make neuronal cells. The other group separated by FEX makes then even much more efficiently the neuronal cells. So yeah, this we could confirm. But whether it is SOX11, this is currently my suspicion, we don't know yet. So what I did not yet mention is this middle group here. So we have genes that go down and genes that go up, which are these genes and those genes. So here we have parasites. These genes fade away. The neuronal genes come up. But there's this very interesting group of genes that is in between. So it is increased during the reprogramming and goes down again. Nogging, SPP1, and DKK1, which are very interesting genes. And so generally, we first looked at these groups of genes in situs, and we see that parasite genes are usually localized at around blood vessels. These neuronal genes are localized in regions in the brain where you would see neurons. And these what we call switch genes, the genes that turn on and then off again, we find them really exclusively in neurogenic or in germinal zones, in zones where progenitors are located. And this is very remarkable, I think. So the parasite gene cluster is really localized at blood vessels, also in the primary mouse brain. The neuronal genes are found in neuronal regions in the developing brain, and these switch genes are really found in ventricular zones. So this somehow suggests to us that during this ASC1 SOX2 mediated reprogramming, we go through some natural neurogenic program, which previously hadn't been really appreciated, that during reprogramming you might go through a progenitor state. And we were wondering whether we can play around with this state a bit. And actually, most interestingly, we see noggin and DICOP1 expressed. And this really suggests that in this intermediate phase of reprogramming, we have dual SMAT inhibition going on. And this is something that, of course, everyone knows can be used to generate neuronal cells. And so we were wondering whether we can modulate this intrinsic dual SMAT inhibition program that is running during this reprogramming. So we add nodal and BMP4 to the reprogramming, and in this way we counteract this intrinsic dual SMAT inhibition. And then on the other hand, we try to modulate the reprogramming by enhancing it. So we help with SP and dosomorphin to even more inhibit, to increase the dual SMAT inhibition. And then we look at the reprogramming outcome, and we can see indeed, this is the non-treated situation, the untreated situation. If we add nodal or BMP4, we reduce the efficiency of reprogramming, whereas if we add SP and dosomorphin, we strongly increase the reprogramming. So there's really this intermediate state where a somewhat natural neuronal progenitor state emerges that undergoes dual SMAT inhibition, and you can modulate that. And not only this, we can also look at the neurons. In the untreated case, you see that these are much less complex than if we add dosomorphin and SP, and in which case you also mature the neurons faster. And also on the transcriptome level, we see that in the case of dosomorphin and SP, we have the same induced GABAergic neurons, but they seem to have proceeded further in the maturation. Yeah, and with this, I summarize also this last part. I showed you that using single-cell transcriptomics on these reprogramming systems, we can identify alternative fates that emerge, not just in this parasite to neuron reprogramming, also during the fibroblast to neuron reprogramming. We find one subpopulation of parasites, human parasite-derived cells, that give rise to induced neuronal cells. The other one is inhibited in that process. And ASC1 SOX2 mediated reprogramming of the parasites unfolds a natural neurogenic program involving dual SMAT inhibition, which can be modulated to optimize the induced neuronal reprogramming. And finally, single-cell transcriptomic analysis can give you clues on how to maybe enhance your cell engineering. And with this, I want to go back to this figure I showed you previously, where we think single-cell genomics and transcriptomics can help you assess the quality of cell engineering in 2D and 3D culture. And I think even more, it can now also give you clues how to optimize your engineering. So For example, you can look at the cell composition of primary and in vitro derived cells, in this case primary as squares and organoid maybe in round circles. You can see that there are lineages that are maybe missing in the organoid, so this is only primary cells here. And then there are cells that you find in the organoid that you don't have in the primary. Those are off-target cells, so you don't want them. And here you can then calculate the similarity, so you have some cells in the primary reference that you don't have, so you have no similarity of any cell to that cluster. And then you have other clusters that you have also no similarity, but those you find in the organoid. So this gives you now a hint. You should try to really make sure this cluster doesn't appear, but you could maybe introduce this other cluster that is missing. Similarly, looking at differentiation pathways, you can do these lineage inference analysis and try to match the reconstructed trajectories. And that will maybe tell you in this scenario, for example, in vitro at the bottom, in vivo at the top, that you have quite a good matching of your in vitro and in vivo differentiation, but at the very end, in vivo, you go into a maturation trajectory that you just cannot achieve at that point in vitro. So your similarity goes down over pseudo-time, and you could then maybe find genes that correlate with that, and it gives you hints for how to maybe better mature the tissues or the cells in vitro. Yeah, and so that's my last message. I want to thank my group. In particular, Gray Camp, who did a lot of the work on the human organoids. Sabina Canton, who and is still doing all this work on the great ape organoids. Of course, that was a collaboration initially with Wieland Huttner's lab, in particular Fahad Bacha, and also in the beginning for the human fetal organoid comparison, and then Felipe Mora Bermudez for the human chimp comparison. Marta was also always involved. Sebastian Ebert, more recently also working on these induced neuronal reprogramming systems, and generally all my collaborators and the funding sources. Thank you.